Maria Avila Arcos, she's an associate professor and coordinator at the International Laboratory for Human Genome Research at the National Autonomous University of Mexico. She completed her undergraduate studies in genomic sciences at UNAM, and later she obtained her doctorate in paleogenomics at the University of Copenhagen in Denmark. She subsequently completed a postdoctoral fellowship in population genetics at Stanford University. Her work has focused on analyzing ancient genomes from a variety of species, including plants, viruses, animals, and humans. And she has also studied the DNA of various present-day and ancient populations from the territory that is now Mexico to understand their demographic and evolutionary history. Her current research interests revolve around how the genomes of Mexican populations, as well as various pathogens that have infected them, have changed over time. She investigates these changes using both ancient and modern DNA. She also conducts pioneering work on the characterization of the African genetic component of the Afro-Mexican population, and her research has contributed to the understanding of the demographic and adaptive processes that have shaped the genetic diversity of populations in Mexico. Her work on ancient pathogens has traced the routes through which some pathogenic bacteria and viruses entered Mexican territory during the European colonization. So it's a pleasure for us to have Maria today. The floor is yours.
Thank you, Xavi. Thank you, everyone, for being here. So, yeah, I think this will be very different from the previous talks that we've seen, but it still touches on very important ethical aspects of the field of, I would call, ancient DNA in general, although I'm focusing on ancient pathogens right now. Well, the study of pathogens and ancient pathogens is the study of humans and ancient humans as well. So we should keep that always in mind. And so this is something I've been thinking a lot about in recent years, and I was finally able to pin it down in some concepts and maps that I will show you throughout this talk. So first of all, I want to start with a disclaimer that this presentation, as I said, stems from many years of personal reflection on the ancient DNA field and its power dynamics. And it might carry some unconscious biases. And my perspective, I know it can have some limitations, so I'm very open to insights from the audience that might reveal some of my blind spots. And the goal is to enrich always the discussion. So when we talk about pathogens in the present, we should always keep in mind that pathogens have been with us for since the dawn of our species. So here I'm showing you a timeline depicting different pandemics impacting humans for the last hundreds to thousands of years. And the impact they have had is shown in bold numbers here. So we have the Black Death, the Black Death, which was caused by Yersinia pestis, smallpox, the Third Plague, etc. So this graph came out during the COVID pandemic, kind of to illustrate that this was not special in the sense that this has been repeated through our history in many years, in many occasions. So as someone who studies ancient pathogens, I looked at this picture and I started to think, okay, what's the, I mean, that it's kind of supportive of we are studying ancient pathogens, right? But what's the impact of these ancient pathogens that cause these epidemics in the past, in the present? What's the impact in the present? So, for example, these pandemics caused by Yersinia pestis, what's the impact today? So I started to pull these maps from websites reporting the impact in the present. And for example, this is the plague cases in the present. It's not even deaths, it's just cases. This is smallpox cases before its eradication in 1979, and this is a map from 1977, where the most deaths, these are cases, were reported. These are cholera cases, and also in the modern era, and this is flu as well. So maybe you start to see a pattern, which is what I started thinking some years ago. First, well, even though plague is the most studied ancient pathogen, it's not a big issue today. And the pathogens that caused the big epidemics in the past seem to be causing the biggest impact in the global south in the present. So I had those thoughts like hovering in my head for some years and thinking, okay, what do I do with these thoughts? And nothing much happened. But then I had the honor to start writing a review on ancient pathogen genomics with these three magnificent scientists. And then I came with this idea of how can we use this information of ancient pathogens and present day global burden of disease. So there is this amazing resource where we can study the global burden of disease in the present. It's curated by this Institute for Health Metrics and Evaluation from the US. So this resource, you can visualize in maps the impact worldwide of certain diseases. In this case, I'm showing you the impact of communicable maternal, neonatal, nutritional diseases, but you can pick different diseases to be displayed here. And these are deaths per 1,000 individuals. So my goal was, and with the help of my brother, who's a cartographer, to kind of visualize side by side what ancient pathogens have been studied and their impact in the present. So, for example, we have hepatitis B. This is the range of hepatitis B genomes, ancient genomes that have been recovered and published. And in this map, we see the impact in the present of hepatitis B. These are other viruses for which there are no current reports in this database, but we have measles, for example, which is becoming very important in the present. And we also see here the deaths caused by measles in the present. And this is the range from when it was studied. Same with influenza, for example. This is malaria, a big problem in globally today. TB, Salmonella enterica, causing typhoid and paratyphoid disease. Lepre, and then Yersinia pestis, as I said, is the most studied, and the few cases, these are just cases, there are no deaths reported from it. And then other pathogens. So this triggered more thoughts. For example, many pathogens that studied through paleogenomics have higher mortality and incidence in the global south in the present. And some of the ones we study don't seem to be lethal or really relevant in the present. So then I asked myself, okay, what's driving this, what I called a symmetry, or this couple of asymmetries? Well, okay, so can we visualize where are the ancient pathogens, from where are the ancient pathogens that the field is studying? So what's the geographical distribution? And for that, I use this amazing resource from SPAM, which is kind of, I guess, a sibling of HAAM. And they have curated data from ancient metagenomics. So I recovered data from this resource. And okay, here I display the distribution of recovered ancient pathogen genomes, the geographical distribution. This is a tool that I'm developing with my brother, where we will be able to see, kind of clicking on each dot, the detail of each sample. So I want you to look at this map, the distribution of ancient genome studies, and where these diseases I mentioned earlier are affecting today. So that's kind of one of the asymmetries I found. So, okay, these are the genomes, the geographic location. Where are they being studied? That was my next question. So this is, again, the distribution, and these are the labs studying this genome. So again, we see this asymmetry in the sense of, or the distribution is biased towards the global north again. So my next question was, okay, how much does the geography of an ancient DNA lab, so the location of the lab, impact the studied sample? Are they only studying like samples from their vicinity, or how does that look? So here what I'm showing is, for example, the samples done in European labs and broken down in colors by the type of pathogen. But, well, you see that the samples done in Europe, of course, they concentrate in Europe, but they cover the entire world. Whereas if we look at the samples done in America, we have fewer from America and fewer from other places. And interestingly, the samples done in Asian and, well, basically this is Asian labs, they do they are processed. The samples are processed in the same country, and this is related to regulations. So I started to see more of the bias, and I identified that Germany is one hotspot for this research. So geographical location of samples studied in Europe, I showed you this before, and if we look at the one done in Germany, well, it's a majority, right? And again, broken down by the types of pathogens. So here I ask myself why, and the answer is complex and it has many angles. But something I came up with while reading and studying about the history of science, I came up with this idea of transgenerational colonial entitlement. So these are Wikipedia pages of, for example, 19th century English naturalists, 19th century German naturalists. And the numbers are large. I think these two are the largest. They have the largest list of 19th century naturalists, Germany and English. So to me, this is, I mean, as a Mexican, I'm growing up, going to school and seeing the role models for science in Mexico and the kind of research is done. Like I would never reach at this point in my career and be like, oh, I want to study samples from all over the world, right? That wouldn't be instilled in me. Whereas I guess in these other countries with this tradition of global exploration and sampling and extractivism, it's kind of transmitted transgenerationally in a way. So then I had more thoughts, and those were that we are not really studying the pathogens with the highest burden at the places where they cause the highest burden, right? Oops, sorry. Sorry about that. And then the labs from Europe recover ancient pathogen genomes from a wider geographical range. And within Europe, Germany is the country where most samples from the widest geographical range have been processed. And we can also summarize this in the classical global north and global south divide. And for example, here I'm showing countries of labs processing samples from the global north. So labs, you can see the list here. So mostly is labs from the global north, studying samples from the global north. And if we see the opposite, labs processing samples from the global south, we still see mostly countries from the global north. So these are just ideas, and what I draw from these are these asymmetries. Our field does not seem to generally use present-day pathogen research priorities, and they exist. Like who publishes these priorities to guide the ancient pathogens we are studying? And should we do that? I'm not sure, but the information is out there. So we could, in principle, do it. And then that our field reproduces historical scientific dominance in the natural sciences of Europe over the rest of the world. But it also inherits the particular dynamics of the ancient DNA field. So it's complex. So I think what I want to draw, the most important thing is that we need time to reflect on these patterns. And so we can imagine how we want the field to continue in future generations. I think that's the most important thing, and not just keep producing and sampling human remains to recover pathogens, because recovering pathogens requires a large sampling of ancient human remains and destructive analysis of them. So I think it's very important to reflect, if we're gonna do the destructive analysis, what's the benefit really? So with that, I will end. I want to thank funding agencies, my group, SPAM, and colleagues and collaborators. And with that, I'm happy to finish here and take questions for the panel.
Thank you very much.